All right, so welcome back to RNA-seq analysis training program, OmicsLogic Transcriptomics. I see that many of you have submitted the project proposals, so we'll review some of those today. And before we do that, let's just take a look at some of the different methods that we covered and where we are right now in the full program. So we start off the program with reviewing the basic processing pipelines. Remember that when you process the data, you want to make sure that your reads are clean from any artificial sequences. And then the important part of that processing is mapping to understand the level of expression of different elements that compose together a gene. Now, today we'll look at some of your submitted projects, and we'll discuss how that is an important step, especially when you're dealing with non-model organisms and when you have some variation from maybe the most studied species used in the reference. So we'll talk about some of those challenges of mapping. For mapping onto reference genomes for well-characterized organisms like human and mouse, alignment-free strategies are also used, and we talked about those like salmon and other approaches. So that is an important part in preparing your data for analysis. Then we talked about the need for normalization of that expression level. And here we talked about genes and isoforms as two ways to look at our data and the need for having a data set that is distributed. Okay, just one second. I'm going to answer David's question here. So David, where you submit your project is on the forum. So let me just show you. Right here on the forum, the first topic is going to be about the project submission. So right here, you see there's a project form. I'm just going to put this in the chat. There you go. Actually, I'm going to put it in the common chat for everyone. Okay, so when you go in here, you'll see there's a form and your details should be filled out if you're using the same email address in the same location. So you should be able to see your information here, and then you'll have these sections to fill out. So we'll take a look at a few examples today just to kind of understand what goes in there. Okay, so we talked about the challenge of mapping. Then we're talking about the challenge of preparing our data for analysis. And this really has to do with exploratory data analysis, where we look at what's data. We want to understand the major trends. For example, principal component analysis could be a useful way to look at our data. You probably have noticed that between when you analyze your data after normalization or without normalization, your PCA is going to change. You will also see your things like distribution of values being very, very different, which has an impact on how you can compare different genes between different samples, right? And we'll take a look at some of these today also when we look at some of the project examples that you have submitted. So then we talked about statistical analysis. And when we think of statistical analysis, we are trying to use some sample of the greater population, right? The basic of statistical analysis tells us that when we have a greater population, for example, the population of all people with breast cancer in the world, and we want to understand whether that population could be studied using a group of people, we want to assume that this is a representative population and provides us with some trends that we can generalize. So it's important to see how we account for all kinds of variation that are not related to the biological phenomena that we're studying. So we want to account for different ways of data preparation and different ways of original material that we have in our samples, all of those kinds of factors. Okay, so then we talked about machine learning and classification with feature selection, clustering, etc. Those methods are really useful when we have sufficient amount of data, right? So when we have only several samples per group, and we only have a couple of well-defined groups that are experimentally controlled, really we don't have such a need for these techniques at the level of samples that we're dealing with. It might still be useful when we want to analyze genes, because we still have thousands of genes to work with. But when we come to a problem where we have many samples, a lot of variability that we don't want to ignore, right? We don't want to assume that we can reduce this natural variability artificially. For example, when we have real human patients, we expect there to be a lot of variability. When we have non-experimental conditions, so sampling from the environment and things like that, right? So we expect a lot of variation. And the usefulness of these methods is to find patterns that will be difficult to characterize from a statistical perspective. So one of the things that I think it's important to remember, and a good illustration here is to show the similarity between clusters, groups in PCA, and differential gene expression. So when you have clusters, those clusters resemble groups that are well defined in the principal component space. But if you take two clusters and you want to run differential gene expression analysis between them, you might not find a lot of genes that show such a difference. And the reason is because clusters are not defined by an up-down regulation. They're defined by a whole pattern of gene expression values across samples. And that similarity in pattern does not necessarily mean up-down regulation. Okay, so then we talked about single cell RNA-seq, and I saw that some of you ran a pipeline. I saw that some of you also ran the pipeline incorrectly. So remember that whenever you have a pipeline of single cell RNA-seq, you have two files. You have the barcode for a sample, and you have the sample itself. So that's just an important thing to keep in mind. And then when you do your analysis, you're really looking at the one sample can contain many, many cells. And so that's really what you're studying, is the variation of a gene between different cells in your sample. And typically you want to look at that variation based on some expected variation between, for example, cell type belonging to tissue, etc. And then we talked about how does all of this apply to individual projects. And so that's what we'll talk about today, because we will see that in projects, all of these strategies change based on the question that you're asking. And so it's important when we look at a project, we think through what are some of the important steps, how do we do the analysis correctly, and what might be some strategies to then apply validation. So what kinds of validation we can do in different examples. Okay, so let's take a look here. We have our program page. I think what was exciting to see on the program page is that many of you have run analysis pipelines. Many of you have completed some of the coursework and tried different types of coding exercises. So all of that, I think, shows that as we approach the stage of defining a data set and trying to apply the tools to this data set, you're probably getting familiar with a lot of these methods and what types of applications there are for the different types of analysis and different stages of analysis. So I think that's good. And I wanted to point out that there are several profiles that kind of stood out from the people that have done exceptionally well in terms of completing many of the assignments. So I've just kind of looked at some of the profiles and selected a few that made significant progress. Doug here has close to 4,000 points, has completed a number of courses. We have Francisco Ferreira, who's got close to 10,000 points, so very close to reaching that 10,000 point threshold. And then we have also Marek, who has been in some of the previous programs as well. And so he has well over 10,000 points. And I think the important reason why I'm showing specifically these profiles is to explain that just from our experience, we've seen that many of the people that join the program, as they figure out their schedule and how to effectively incorporate both the synchronous learning and the sessions that we have, by this time, to be effective at addressing the challenges in a project that you will be working on, typically you need around 10,000 points. And what that basically boils down to is if you do Transcriptomics 1, 2, 3, and 4, and Introduction to Bioinformatics, with the required assignments within those, that basically puts you at around 10,000 points. So that's kind of what you can see here. And I think that, you know, for those of you that are interested in continuing further and kind of getting the most out of the next couple of weeks that we have remaining in this program, which is to work on the project, the 10,000 threshold, first of all, kind of qualifies you for a certificate of excellence, but also we will be trying to help those of you that have put in the effort in the beginning of the program to make the most out of the remaining 2 weeks and help you get your projects done well. So please keep that in mind, and please use the rest of this week to complete as much as possible and complete your profile with the project submission so that we can also help you move along in your project. So again, just to kind of remind everyone, we have this online coursework. You should have access to a lot of the online coursework. And this coursework will introduce some of the topics that we discuss in the sessions, helping you practice independently and helping you review the concepts and the terminology that we are discussing. Now, overall, I did this review last time already, but just to kind of remind everyone, statistical analysis, when we come to this stage where you're trying to think of different types of data sets, in many cases, you will find different numbers of samples and different level of variation as you look through these projects. So it's always important to look at how exactly was the data collected. You know, did they breed the same genetic strains of mice, and then they were homogeneous in terms of their age, etc., sex, and then designed an experiment, you know, with a knockdown experiment with a specific gene that they wanted to study? Or did they go out, sample a real population, just kind of, you know, providing us with some metadata, with a lot of that metadata probably insufficiently describing the real variation? And so the different approaches that you might want to take will reveal different types of patterns in the data and different relationships, right? And you can really combine the two, but that requires first an exploration. So you need to understand the technical variation in your data. You need to understand the variation at the sample level. And then if you want to extract some common trends, it might include removing some samples from your data and kind of using normalization to make sure that the assumptions you will extract from the analysis are, you know, realistic in terms of your biological or biomedical interpretation. Now, in many cases, when we look at the gene expression patterns, remember that the variation here is at several different levels. So we talked about how the variation could be at just the level of expression, but level of expression is not the full story. And so we'll take a look today at one of your project examples where I think this is going to become clear. So after we have established some pattern of variation, the details of what can we do moving forward with this gene really make it much more interesting, and only maybe the beginning of a real biological question that you might have. For example, if you have a gene that has the same level of expression in different samples, but has been assembled from different numbers of exons or exons in different orders, that can change the function of that gene. You might also have a gene that has mutations in it, and you can study that also based on the sequences that you have from your gene expression data. And you can also ask additional questions about the role of that one gene that you're interested in the context of biological pathways and function that could be inferred from pathway analysis and gene ontology analysis based on common functions. So I think that overall, thinking about where do you end up after you've gotten your table of expression and you've identified some interesting patterns, in some sense, that's where the project begins. So preparing the data, making sure that you understand variability, asking questions using statistical methods to confirm or disprove your hypothesis leads to a number of additional questions that you might want to continue asking about the data sets, and then leveraging databases, but also thinking about how could you validate this or how could you define this using biological experiments, or maybe validation using other publicly available data sets. So that's really the direction there. And we talked a little bit about some of this variation last time. So two options for those of you that want to move on and explore how all of this works in other projects. We have some data sets that were already compiled in kind of tutorials that you can go through. So these are the projects that are made available. The most recent ones, I think that could be useful, and one that is almost completed and we hope to release for all of you to try as this program is going to be drawing closer to an end, are the Ebola virus. We have a project on liver cancer, and there's going to be a project that's coming out for COVID-19. So these projects is a good way to finalize the program. They all are based on a publication, on a data set, and then you can do some variation of the methods or some additional data that you can add to this project and really get into the topic in a deeper way. So again, the transition to go from learning to then understanding how it applies to a research question is really applying these methods to a data set where you can find some interesting patterns and then interpret it, comparing to what the authors maybe propose, and asking some additional questions that you think of how could these questions be answered using some other data sets or some additional methods. We do have a number of previous projects that were recently completed, and I think that these are projects that are good from a standpoint of a project that was completed in a short period of time, and some direction. They provide some direction for those projects that could be done by you during this program. So I'm just going to post the link here. Okay, one sec. Okay, so here you can find some of those project examples. And in the project examples, there's also a little tutorial on how to really go from a general project to a project that is more interesting. And here are some examples that you can look at that people did, including some projects using RNA-seq, and some of the recent ones that are not related to RNA-seq necessarily. So you can really look through some of these projects to think about what you can do in your own project. Okay, so are there any questions about projects before we move on to some examples from what you have submitted? So are there any questions about, where to find data, how to, come up with a project title, or how to, develop an analysis strategy for the projects that you're thinking about? Okay, well, let's take a look at some of the projects that you have proposed, just to kind of give a review. The first project comes from Francisco. Okay, where to get data? Yeah, so we will take a look at that today. Francisco, is it okay to use your project as an example to kind of go through this together with everyone? Okay. So Francisco submitted a project for mosquito response to Plasmodium infection, so malaria infection in mosquitoes, and he defines here the problem: the process of vector invasion leading to the production of infective parasite stages is physiologically complex. Mosquitoes have to fight off the infection, while Plasmodium parasites have to overcome such defenses for a successful transmission. And so Francisco here says that there's a link to some data and a reference publication. So Francisco, would you like to kind of expand on what do you mean by this complex physiological process, and what exactly are you interested in? Do you want to just say a few words about that?
Yeah, so how do you want me to do that? Do you want me just to briefly explain? And this picture you have here is pretty helpful for that, if you want.
Yeah. So that would be great if you wanted to say a few words for that.
Yeah, so as we can see here, the parasite has to invade the mosquito, and the parasite has to develop into kind of a complex stages. And now in the midgut of the mosquito, we can see here in the pictures a lot of genes are involved in the mosquito, on the mosquito side of things. And then the parasite has to burst out in the midgut of the mosquito, and then it has to go through the whole mosquito body, through the hemolymph, is a kind of the insect blood. And this insect blood also has some defense cells, kind of like our white blood cells. And then after going through all this journey, the parasite has to find the salivary gland that we can see here in the picture, and it has to cross the salivary gland, and they stay there until the next time when the mosquito take a blood meal. So we would definitely, at the end, parasite has to go from the midgut to the salivary gland, and then to be injected in the blood meal. So I want to see which genes are responsible, which genes are kind of turned on and off or differently expressed during this process. But I'm interested in the mosquito genes. They are differently expressed, and then to see how the mosquito try to fight off the infection, and also there are some probably the parasite also has to kind of manipulate and to escape the mosquito response. So this is more or less what I'm doing now.
Great. Yeah. Thank you for that explanation. So I think really a fascinating aspect of infection where we see tissue tropism. In other words, that the pathogen can replicate or can invade certain tissues more effectively than others, and different stages of the development of the pathogen happen in different tissues. And then in humans, it happens differently, infects different types of tissues. And then also we can see differences between infected and non-infected mosquitoes, right, as described in the project reference publication. So let's take a look at this reference publication, which is right here, and we'll kind of see, first of all, what the data is, and then we'll also look at what is the proposed analysis that they did, and also talk about, you know, what you plan to do with this. Okay, so this is the reference publication: gene expression changes in the salivary glands of Anopheles coluzi elicited by Plasmodium burgei infection. Malaria is a devastating infectious disease caused by Plasmodium parasites transmitted through the bites of infected Anopheles mosquitoes. Salivary glands are the only mosquito tissue invaded by Plasmodium sporozoites, being a key stage for the effective parasite transmission, making the study of Anopheles sialome highly relevant. So just briefly skipping over to what exactly did they do? What they did was they extracted RNA from mosquitoes, prepared it for sequencing, and then they also looked at some additional elements of the differentially expressed genes, right? So I think that was an interesting approach. So I tried looking through this publication and kind of, you know, building this, this schematically over here. So they prepared the data basically from infected and uninfected mosquitoes, as far as I understood. They did a differential expression and then looked at how those differentially expressed genes could explain some of the biology using gene ontology annotation, and found some interesting differentially expressed genes. And what they were specifically looking for are the genes that are involved in the process of infection. And they discovered one particular gene that belongs to a group of proteins. It encodes this group of proteins that could be further studied for its functionality. And so that's where they turned to an additional set of methods for multiple sequence alignment. So aligning the sequences of the genes that they found in these mosquitoes, and then also looking at how these genes in general could be organized by different clades, how evolutionary they evolved to have some specific function, because these genes are present in different organs and different species. And since in the mosquitoes we have this specificity by interacting with proteins from this pathogen, they could ask the question, what structural characteristics of this differentially expressed gene that seems to play a major role in the process of infection could be inferred from this, you know, structural analysis. And they also did this validation study where they knocked down this gene to make sure that biologically that is, you know, evidence for involvement in the infection. And so I think an interesting study. Now, in this, they, you know, Francisco, you proposed to take the vector-based data, even though they provide us with a data set from Array Express. Is there a reason why you wanted to take the data from this database, or and have you looked at the data that they actually use in this publication?
Yeah, actually, I didn't look at this database. They're referring to this ArrayExpress. I didn't look at it. I was just not looking in back to base some other studies that did more or less what I want to do. So I didn't go into more details or the data that they released or where they provided data. But that's definitely going to be very helpful to dive into this one. For me, it's very
Okay,
new.
so
To me, it's new this database.
okay, yeah. So they mention here that they deposited their data in the ArrayExpress database under accession number EM tab 3415. So if you go here, they provide you right here with a link to the data. And here, let's see if they show us some, because I think the database provides some aggregate information from multiple studies, right?
Yeah. The vector base is cool because we can it's very user friendly, and we can run some of the analysis straight from the platform. So it's a kind of and then they collect also other many studies in the same platform. So is good because of that.
Yeah, so I guess one of the challenges in your project description would be to define what specific analysis would you want to do? Because as you mentioned in the submission that you did, you want to pinpoint mosquito genes that are differentially expressed during Plasmodium invasion. So I'm not sure specifically as you want to look here for these differentially expressed genes, how would you say that these genes are differentially expressed during infection, or during specific stage of infection, or whether they were captured from some specific tissue like salivary glands or another tissue? I haven't really seen here how you could use that analysis approach, as opposed to maybe use this specific database more for validation, right? So once you have found the genes of interest, whether from literature or from analysis of raw data, then you could have some downstream validation steps, right? Let's find these specific genes like it suggests here, and see what other types of variation are known to be present in a variety of different studies. See what I'm saying?
Yeah, that's correct. Yeah, that's what I'm doing. I actually have my some data yet already, so I'm kind of that's what I did. I did all the pipelines, and then I just uploaded the genes that were differentially expressed into this platform, and then they kind of tell me which which metabolical pathways those genes were involved, and then they can also they do some gene ontology analysis as well using this platform, connecting to the another servers that does gene ontology. So yeah, you're correct. I This is more useful for downstream analysis after we already have all the pipelines up to have the read count, the read count table. And then we go to this platform.
Okay, great. Yeah. So I guess what we want to specify in the original project is what is the raw data set
I see.
that you would, yeah. So it says link for raw data.
All right. Yeah. Okay. I see. I see your point now. Yeah. Definitely. Yeah. Definitely is not the raw data. So I would say is my data for now.
Okay. So yeah, so I think that the project itself looks very interesting. Finding differential genes, what I would suggest is, your project summary should include these steps, just like are defined right here as an example from this reference publication. So
I
you
see.
want to say, when I do my quantification, these are the types of samples that I have. These are the groups that I will compare. This is why I think these groups are meaningful based on number of samples, based on what tissues they were from, etc. Then you would say that, let's say you do differential gene expression and gene ontology. Before jumping into the specific questions, you want to just understand the general trends, and you want to understand, whether the biological function maybe is confirmed from literature, right? So in literature, we know so and here we see the same thing, right?
Mm-hmm.
In terms of what they did here with this gene, similarity with other types of species, and then structural analysis even, I think that's a very interesting approach. I'm not sure if that's the direction that you want to go, but if you could kind of prepare a graph of all the steps and what those steps will produce, and then explain how that will lead to these specific genes, and you would want to then, maybe what else are you going to do, and how is this different from what's already known, right? What is some new outcome that you're looking to gain from the study, as opposed to what's already known and described in literature?
Yeah, that's because also what I'm doing, I wasn't clear here. I'm using I'm using more a bird malaria system from Hawaii, so which is
Mm-hmm.
pretty a little bit different from human malaria. So I'm using the human malaria system kind of to understand, because no, of course, way more information about human diseases than bird diseases, and especially birds from Hawaii, which is a big problem there. So my question is more about that. Yeah, and then I'm looking for other studies that can kind of that can compare to see. And this structure comparison is interesting because for the bird malaria, the vector is totally different. Is a Culex mosquitoes, another group of mosquito. So it would be interesting to see if those genes, they are differently expressed. The function is conserved, between those two different mosquitoes, the human malaria and bird malaria, and see what are even the phylogenetic relationships between those genes, or even the full metabolic pathway response, if they would be more or less the same way, they would fight the parasite more or less in the same way, even though they are very different parasites and very different mosquitoes.
Mm-hmm. Mm-hmm. Yeah, so I agree. Those sound like very interesting questions. Um, as you define the specific questions and you get some preliminary results, then you can think about, how do I illustrate this? And if you start from just kind of giving an outline of all the analysis steps,
Mm-hmm.
and then thinking through whether the data that you have, has sufficient number of samples and things like that. And then also when you're thinking about your outcomes, maybe you could define in terms of literature, where are those most interesting questions? Is it the difference between birds and, humans? Is it the difference between the different mosquitoes? Is it, the difference between specific genes? So, obviously there are many questions that could be asked. What are some of the most interesting questions, and
Mm-hmm.
how would you define those most interesting questions in the context of the data that you have to, answer those questions?
Yeah, I will do that. I work more on this on the summary, and I can update, maybe today or tomorrow, and go from that.
Perfect. Okay, well, thank you. I think,
Mm.
yeah, very interesting question. Does anybody else have any questions about this project for Francisco? Okay. So if you do, then put them in the chat. Another interesting project that I wanted to turn to is the project submitted by Doug. Doug, is it okay to discuss this project kind of in general details here? Do you want to say a few words to just kind of introduce the project and what you're trying to do with it?
Sure. Can you hear me?
Yep.
Okay. So I have data. This is a heat shock study on cutthroat trout, and which is the one you can see down in the little right where your marker is, just to the left of that. They are sister species to rainbow trout, which is a fortunate thing because the rainbow trout extremely widespread worldwide, uh, um, economically important species. So there's a lot of molecular tools, including a genome that's been done for the rainbow trout. That it's a sister species, so we can map onto that. It's important to have that genome because these have been through two more genome doublings versus mammals. So it's a very, very different complex genome. And a number of studies have been done. It's in the genus Oncorhynchus, which is the Pacific salmon. So a lot of comparative sort of studies that have been done on related species. They're very, very cold adapted. And so, 20-degree normal room temperature is warm for them. 3-4 degrees more than that, and they're going to die. And so what we did was heat shock them. It's a pretty standard protocol where you ramp it up. It's an acute heat shock for a couple of hours and then sample them versus control. I have data from one of the things that makes this different, I have data from 5 different populations. And hold on just a second. Sorry, my wife's tea. And so I have data from 5 different populations, 3 different subspecies, and they were reared in captivity, heat shocked for 2 hours, and I took RNA from gill tissue. Gill's really on the front lines of dealing with the environment. So and that's what many of the studies have looked at. And so the idea is of course, here in Colorado, these are very high altitude species. There's not much higher they can go because the water gets too sterile. There's nothing there for them to eat. It freezes in the wintertime. It only takes a day of really warm water to kill off these populations. One of my 5 populations is known to be highly inbred. And that is something that makes it really interesting in terms of the way those really don't deal well with stress. And so that's what we're looking for, is at what point, so treated the same way, there's a sequence from the heat shock response to pretty quickly getting them overwhelmed to where you start seeing apoptotic pathways kick in. And working with Colorado Parks and Wildlife, we're trying to determine which of these populations has genes, and we know a little bit about the phenotype, and we're trying to just see if we can find biomarkers or something like that.
Okay, so biomarkers that differentiate between their tolerance towards heat.
Yes.
Okay, and so can you describe a little bit what is the raw data that you have? So you say here fish from 5 populations representing 3 subspecies. So maybe what would be most useful, just kind of, for everybody to think about this, what are the specific comparisons that you can make that will show, one, I'm assuming that you have 3 subspecies, right? So 3 subspecies, they would have some genetic variation, but then 5 populations within the 3 subspecies, right? So is this 2, 2, and 1, or, how exactly does that work? And then you have different temperature levels, and right, and then, oh, okay, well, yeah, so you have 10 fish from each population. So these are the 5 populations. So how does that divide into the 3 subspecies, I guess, right?
Right. So there's 3 of the populations are of the same subspecies. And then there are 2 other subspecies that are represented as well. One of them is Yellowstone, which is just more widespread and more highly studied. So they're there for that reason, as well as others. They seem to do better in a little bit warmer water. One is our Colorado State fish that is highly endangered and highly inbred. And in terms of, so that, yeah, 50 samples of RNA, 5 populations, 5 controls, 5 that were heat shocked at 10 degrees. They were all reared together, so they're all of that is controlled. And I've seen done a preliminary analysis of, so I've got the, as you know, I've got the table, the DE table on them, and I've looked through that by hand and pulled out some of the genes that caught my eye that were highly differentially regulated, a lot of heat shock proteins, not surprisingly. And on a PCA, in the first dimension, it pulls of course, it pulls the heat shocked ones away from the control ones, which is nice. That was the experimental design. That's what you expect in the first component. The second component, what I was hoping to see and did see, is that it pulls that inbred population away. And we know phenotypically those inbred ones, they don't grow well. They just have they have jaw deformities and various other sorts of evidence. That's one of my interests, is to try to pull from this data. I've got a review that has a section called Transcriptomics signature of inbreeding depression. That's one of my interests. Not exactly sure how you do that. I think it involves SNP analysis and then trying to look for some sort of population genetic parameters in terms of heterozygosity and so forth. But so that's one of the directions. So I in doing our courses here, beyond the principal component analysis, I haven't seen this so much, but it occurred to me that using the some sort of classification analysis to compare the different populations might be a useful way to go. I'm not sure, but that's something that occurred to me.
Exactly. Yeah. So let's talk a little bit about this analysis. So here we have, I'm just going to move this for a second. So here we have your pipeline, right? And so the first thing that we should mention here is one of the challenges is actually having a non-model reference organism, which has, many differences than you would standardly work with a mouse or a human genome, because you have to map on this genome. The genome has to be indexed. You have to have the right GTF file, which has to match with the specific names of genes and the specific positions on that genome. So that's one of the challenges that you wouldn't experience with your standard model organism study. And that's why you can see here the pipeline is actually mapping on genome and then constructing those isoforms and preparing a new GTF file that would then translate into transcriptome quantification. So I think that's a good illustration for where you would not just go through your standard quick pipeline. So that's number one. Number two is you have then variable levels of mapping. So this is an output from most HT-seq and HiSat and Bowtie 2 will have this output where they show you the overall mapping between different samples. And this important step for quality control would help you understand whether there are some outliers, right? Like YC4 right here has a 74% as opposed to most of them having about 80%. So, depending on how accurately you want to look at the data, you would expect that the quantification normalization, for example, if you use FPKM or something like that, would actually account for that, right? Because this number of total reads aligned to the reference is going to be incorporated in FPKM. So another consideration here. And then finally, as mentioned already from the PCA analysis, right, we can see that just taking this data and looking at it from principal component analysis perspective, we do see some interesting control samples over here in the heat shock group. So what was your conclusion about that?
Conclusion that the control samples are in a comfortable temperature. They're at about 17 Celsius, which is pretty optimal for them, versus the others that are really uncomfortably heated up 10 degrees more.
But why would they appear here in this? This is the heat shock group, right? So why would these
No,
control
that's the
samples?
control. Those are controls.
These are the controls
Yeah,
because
the
these
C,
are.
C is for control.
So MCH, what does MCH stand for?
That's a different population that was heated up, heat shocked.
Right. So these are MCH, NH, and TH. So these are the heat shock. All the ones that have H's are right here, except for these two. And these two are the YC1 and YC2. Do you think they might have been mislabeled? Do you
No,
think
you've got in red down at the bottom. So yeah, why the red one is
yeah.
Red,
Right. So all the red and orange are right here, except for these two, which are blue. So these blue ones are, the rest of them are here. So all the other YCs are blue, and these are these guys right here.
I see.
So I think these are unexpectedly in the heat shock group. And I think that the reason for this could be mislabeling, but I'm not sure if that's possible.
That's not what I had seen on the previous principal component analysis.
Well, okay, so this is just something that I wanted to highlight as an important step in your exploratory analysis. These things happen, and they happen a lot. So people do mislabel, contaminate samples. Maybe something happens at the stage of pooling together samples for sequencing. So all kinds of issues can happen. And I think it's good to always be mindful that sometimes, these issues can creep in. And obviously, when you're going to do your downstream analysis, this is going to interfere with your interpretation. Now, many things are possible. Maybe, the PCA picture here is wrong, right? Maybe the wrong columns were selected. You can do that yourself just to validate that everything is done correctly. But let's say if it is done correctly, what would be the ways to overcome this problem? The first one is you could just throw these samples out, right? So if you throw them out, and one good way to validate that that is a mislabeling problem is if you throw them out, could you improve these numbers right here? So the variability, right? This is the best way to kind of turn something that is a cloud of points and do a projection. So if you throw them out and the variability within each group is reduced, then you should expect to see these numbers to go up. And typically, I mean, this is already pretty good. You've got over 40%, over 40%, is considered to be a good representation of your data. It's not the best, but that's okay. But that would be one of the ways to validate. So if you throw away some stuff and then you have improved variability. The other approach is to try and understand, what other things could be different about these two samples from the rest of the group that they should be in. So just do a PCA for the controls, or just take controls you have here, YC and YH. I'm assuming that Y and Y are related.
Yes.
So, if you do PCA just between the Ys, do you see a clear difference between controls just within the Y group?
Mm-hmm.
Okay, so these are just some considerations.
What I had mentioned to you, I just want to point it out since it shows up here. If you look over on the left at the controls, at the very bottom, those open circles, yeah, those are the inbred ones, and that's what I had seen before, that they all cluster on that dimension at one extreme end. And so that's been one of my interests, is to compare controls against each other.
Yeah, so that is, I think the other point. So first of all, Francisco here is saying mislabeling in PCA is pretty common. Done that, had many heart attacks because of it. So I think this is something that, definitely you could redo this and just try and see that, what you are seeing is what's expected, and then do all kinds of comparisons within specific groups, just controls, just Ys, just specific types of comparisons. So the second point you brought up in your explanation of the project is you're saying that there's a lot of this, natural variation because of inbreeding. And you mentioned that, this could be because of mutations. This could be because, other issues. We talked about this before, alternative splicing. It could be, right, different types of exons together, even though the gene is expressed. But the first question would be, I mean, is there any level of expression difference between the different control groups, right? So if you forget about the heat shock for a second and you just take the control, like you mentioned, there is already some visible separation. What if you just do PCA of these and you also ask the question, what are the differentially expressed genes between this group that is inbred and the rest of them, biologically speaking, maybe mapping onto pathways or something like that. The other thing is that, this, I am not sure whether this was done before or after normalization, but as you know, in PCA, you can do scaling, you can do normalization, you can do normalization per group, right? So there are many different things that you can do to try and work with just kind of your exploratory data analysis step. And the last thing is you mentioned biomarkers related to heat shock in specific populations. So I think this is a good classification, as you mentioned, with feature selection approach, where you can say, I've got 5, or, maybe because you only have 50 samples, you don't want to have too many classes because ultimately you need some training data, right? So you need to think here, what is the best way to organize your samples into these classes and divide them up into meaningful groups for training and test data? Because typically training and test data is separated something like 70-30. So if you have 50 samples, how many samples per group can you have to have enough training and enough test data samples to really, get some consistent markers that would be more generalizable, right? I think that's the main challenge here, just because the number of samples is not huge. What do you think about that?
Yeah, the inbred ones is just one of the populations, and I had thought about various different combinations of the others as control in terms of the control groups. Some of the others could be lumped together to compare. That still leaves me comparing against what I've got is those 5 that were. I don't know if that's enough. If I use the
So
others
so you have 5 inbred controls and 5 inbred heat shock,
right,
and then the rest could be grouped together.
but yes,
So you would ask the question, you would have 4 classes: inbred control, inbred heat shock, the rest control, the rest heat shock.
yes,
And then you would want to select genes that show this kind of classification and good separation between these classes. I mean, I think that's reasonable. You should probably try that out. Harpreet, are you on the call?
Yes.
Okay, so maybe this is something that we can set up a call and discuss if
Yeah.
there are any questions about this. Okay, but I think overall, this is a very interesting project, and obviously you've been working and thinking about this for some time. And if you have some additional questions on, how to do anything more specific with this data, I would be happy to help with that. And Harpreet obviously is very experienced with the classification, biomarkers, and all of that stuff. Okay, so I also wanted to, and thank you for sharing this. I hope this was useful for everybody else to kind of think about the issues that might come up in some of the other data sets. Does anyone else have any questions for Doug and his project here? Okay, well, the last question here was about where to find data. And so Harpreet, if I could pass it on to you to show two things. So first of all, just in general, how to find data on NCBI. And two, once you find that data, how can you use the SRA table to prepare a pipeline directly from the SRA data set to run the processing pipelines?
Okay, sure. Ilya. So just let me share my screen. So first, how we can actually download any data set or search a data set in the from the public repositories. So one of the major public repository from where you can find the data, that is GEO, or Gene Expression Omnibus. So here you can search for, if you're interested in a particular, you have a particular research interest. For example, if you have an interest in COVID-19, so you just need to write your query. And then on the right side of this interface, here you will get this option, top organism. So from here you can select on a particular organism that you are interested. So like here, for example, we are interested in Homo sapiens. Then from this on the left side, here you will get this, you will find this option, that is study type. So if you just go to the customize, so from here, you can actually select a particular data types. For example, what kind of data that you are interested. For example, we are interested in high throughput sequencing data. So I can select that particular option and I can unselect all other options. So you can choose, if you're interested in overall any high throughput sequencing, then you can also select if you are interested in some non-coding RNA. So you can select that option, like non-coding RNA profiling by high throughput sequencing. Then apart from that, there is array options is also available. So you can select a particular data type that you are interested. So then we can just click on show. So from here, it will provide us this option. If you click on this, we're able to select only those studies which actually contain data from public report that is associated with the high throughput sequencing. So here, from here, you will find some particular study and you can focus on that. So for just to demonstrate to you, I have selected just one data set. I'm going to search its GSC ID. So here I have one GSC ID. So I already gone through one publication. So like here, large number of data sets are here, and you can go to each of these data set. So like in which kind of data that you are interested. From here, you can actually select the studies based on the number of the sample, that is from high to low number. So for from out of which I have find this particular one study, sorry. So I can share this GSC ID with you. For example, I have selected this particular study. So how you can go to the study, if you select on one study, so you can directly direct it to the complete study, like here. So here you will get exactly what they have done. Like most of times a data set that is submitted at Geo, most of times it is already published. Sometimes it under the communication, but most of times it is published. So how do you know exactly which paper or which study is associated under this citations? Here you will get the complete publication research article that is associated with this data set, and you can go through exactly what the authors have done and what still we can do with this data, right? So then here under the summary, they usually provide complete info, like they summarize the information what they have done. Like in this particular study, they have taken the cell line samples and then treated with some inflammatory drugs, right? So they have treated that, and then they have tried to find, they have took the different types of sample and then treated with mainly two in their study they have considered two types of samples. One is from the human and other from the one other organism, like here, yeah. This is Ptoris furo. So how you can actually download a data from the public repository. So most of times under the supplementary file, authors have usually provided already processed data. So from here you will get this information, like readcon data. They have already submitted, providing or submitting this data at this particular public repository, which is already processed. So in this case, they have already provided readcon data. So you can download that supplementary data and perform your any analysis, like you have performed TCA, or you can perform machine learning, depending upon what type of analysis that you are interested, or you can perform differential gene expression analysis. So this is already processed data. At least they have read count value. This is like already this is not a raw read. So beside, if you are interested in a read count data, if you are interested that you want to process your own data, so beside under the relations, here they will provide the link to other repositories where the data particular this data set associated with SRA or by this bioproject. Then this particular series matrix file under this download family usually contain clinical information of the samples, clinical, or we can say demographic or different characteristics of the sample. So under the series matrix file, you will get that information. So if you're interested in or you want to process your own data, so you can directly go to the SRA. So here we will get this link. For example, here I am just clicking on this SRA. And if you want to process your own data, like you don't want it, the sample should be I mean you are not interested in their processing pipeline, so you want to process your own data. How you can obtain that? So if you this is we are directed to the SRA's sequence read archive. So this repository, usually this sequence read archive or SRA, actually contains raw reads. So if you look at even a single sample here, just let me just show you here. So if you're looking at this particular single sample contain like 3 GB, right? So it is already 2 GB data, 1.5 GB. So like if you have 110 samples in this particular study, so it's like you need more than 200 GB on your if you want to process this data, you must need lot of space and then computational power. So it is like impossible to process this data directly. So what we can do of this if you go to this send to option, so you just what you need to do, just select file, and then under the format just select run info, And with this, we will able to create SRA run table. That this particular SRA run table only what it contains. So let me just show you here. So this is a very, it contain only the information related to the samples. So this is what is the SRA run table. So for example, I'm just taking only 3 samples. So here we know that it is a single end sample. So then we have also know that if you are interested in particular human sample. Okay, so already these are the Homo sapiens samples. So for example, you can select particular sample that you are interested. For example, I am interested in these first 3 samples just to show you an example. So how we can process this data. You can select all the sample that are required in your analysis, you can select all. So otherwise you can select a particular sample that on which you want to perform analysis, right? So you can, once you have this table, so I'm just copying it and paste it in the txt file. So another important point, whenever you doing any analysis on T-Barnford platform, make sure your file should not contain any blank line at end of the file. For example, here I just pasted, and again if I look at this file, so I can look at this file here, I have a blank line. So it is important that I should remove this line. Now I can save this file. So I'm just saving it at desktop. So I'm just taking these 3 samples. So then we go to the T-Barnford server, and let me show you how we can actually directly take this SRA run table to generate our own gene expression table. So if we go to the T-Barnford server, so and here I need to click on this RNA-seq. From here I have to select, so I have to select Homo sapiens. And then these are the FASTQ file. It already mentioned I am taking already genome model genome reference genome because we have. So otherwise we have to submit it. So then there is a single end or pair end it asking. So all, as I have already checked in my data, these are single end reads. So then we can go to the file upload, and here under the NCBI run table upload, here we need to provide only that file. So I'm just uploading this file. And once we upload, then we can go to the group selection. Just group selection we need to perform in that case if you want to perform differential gene expression analysis. If you are not interested to perform differential gene expression analysis directly, so you can perform it later. So we can hide it and just cancel no group. Then we can go to the start. Now I can perform pre-processing step, that is PCR clean and Primomatic, that is one to remove the PCR duplicates and adapters. Then I can directly just run this gene expression table. Then I get I have just after the pre-processing I need to map this on transcriptome. Next I need to generate a quantify expression values. So for that I can choose option from here. So I can choose FPKM value, RPKM value, or expected count. For example, I am choosing only FPKM value. Then we can end this pipeline. So this is how we can directly upload data from NCBI, RNA-seq. So, and run our pipeline. So this is the one way. Once we run the this pipeline, and we will able to get a gene expression table. So the other option that we have, this is if you have only raw sequencing reads, and then you can produce your gene expression table and can perform any analysis. Then beside we have this option, right? Sorry. So this is already read count values. So we can take these value, and let me just show you. Let me download it. So I have already downloaded this file. Just let me show you that exactly what this file contains. So here I have already downloaded this. So now I can open this file and with so I'm just opening in the notepad, and I can show you in Excel. Let me just paste it in the Excel. So this is already, we can add ID because this is missing from this file. So if you look at this file now, this is already a gene expression table. So now you can perform any analysis, like these are read count value. You know, if here in this data, you can select particular samples. If you are like this is already treated with a multiple drugs. So if I can go to this series 16, I can look into exactly what these samples are. So if I'm looking at this, so this is like from series 16 because I have already checked this data. So let me just highlight it. So this data is actually already processed by the authors. So we can directly use this data because this is read count data, and that once we have read count data or the FPKM values, so we can perform any analysis on the read count. So if I select this data, and when I have gone through this sample, there are 3 samples which are mock. Then there are the next 3 samples that is actually on SARS-CoV Rux treated, that's ruxolitinib drug, and then we have untreated drugs, untreated samples. So we can perform any analysis once we have this data. So beside, we can process this table further. Like I have actually processed this data by I have removed all those genes which contain 0 value across all the samples. So now if I go to the I have prepared a file with these samples, these 9 samples, and I have also removed all those samples which contain 0 value across all the samples. So I have considered only one drug treated data. So you can consider any of analysis that you want to perform. So for example, here, if I go back to the tbioinfo server, once I have that table, so I can go to the area of analysis, and I can execute unsupervised machine learning. So if I upload that file, I have already prepared one file from that table, and let me just go to that. So I have already prepared this file which contain mock, SARS-CoV-2 and drugs treated samples. So then I can go to the continue. So I have just selected those particular samples in during the preparation. What I have done, that I have removed all the genes which having a 0 value across all the samples, and then I have selected only 9 samples which are actually related with the drugs treated. So then we can go to perform PCA. And we can perform this particular analysis. So it will take 1 or 2 minutes. You can perform hierarchical clustering to check the pattern. So whenever we have actually gene expression table, so what should be our first approach, that we should visualize the pattern within a data. So that will actually help us to understand what are different patterns. So it will take 1 or 2 minutes to complete this pipeline. So I'm just waiting. So whenever this is very important, whenever you have gene expression table, so you must check that distribution of the sample within your data. So for that, one of the best approaches principal component analysis. So usually it will actually explain you how many types of samples are there. So you can, I mean, this is already running, so it will take a bit of time. So is it clear how we can actually download a data from the public repository? Just let me know that. Is it clear to everyone actually how we can download data from Geo or the SRA? Because here my goal is to actually to show you what kind of data that is available in the public repository, and how you can download and directly perform analysis on TBioInfo using these various tools that are available on TBioInfo server. So this pipeline, because RNA-seq usually take a much more time if because it is going to process raw sequencing reads. So if I go to this PCA, and if we just go to this plot first. So if you look at this, I have not normalized this data. So if I'm looking at this, in out of these 9 samples, if you look at, there are these 2 samples, then these 4 samples, and this is 2 samples. So you can at least visualize there are 3 different type of samples are there. Although this particular sample is getting mixed with this. So one sample seems like this blue color. If you look at all these samples, this blue color sample is mock sample. It looks like it is outlier. So you can look at by looking at even just at the pattern within your data based on the PCA plot. So here, if we look at least we are getting that this data might have 3 different types of a sample, 3 different categories of a sample. So like here it looks like, then you can perform also hierarchical clustering based on all the genes. Then beside, you can, if you have already this raw read count data, you can also perform differential gene expression analysis between 2 classes. So this is how you can directly take a data set from public repository, then perform any analysis on this using different modules and tools that are available on TBioInfo server. You can perform differential gene expression analysis. You can perform hierarchical clustering. So you can then under the utility option, you can perform normalization on this data. Then if you have multiple kind of factors and conditions, so in that case, you can also perform factor regression analysis. So if you have any question, specifically how you can I think I have shown you the one link, and the other link that is actually usually associated with your Geo is also provide the link to the bio project. If you are interested to download the bio data from the bio project, you can also direct it to this. So here you will get all the information related to the bio samples, and you can take and go to the publication as well. So this is how you only, if you just search the Geo for with your research query. So Geo contain every type of information. It's not like it is dedicated to the cancer or something. So it is dedicated to the every single resource. So beside, if you are interested in, like if you are interested in mutational kind of analysis or the sequences. So if for that, there is NCVI virus. If you are interested in want to check the mutational analysis for specifically if you are interested in some virus, so then this is the one of the major resource, that is NCVI virus. So here we can search, for example, if I want to check what are the changes or the changes are taking place. So for that I need actually sequencing data. So how can I download sequencing data? So I can search by the virus. For example, here I'm interested in coronavirus. So first, once you have gene expression understanding, then you can perform analysis at mutational level. So from here, you can select a particular geographical region, like if you are interested only in Asian or African patients. So then you can also select particular virus. For example, you are interested only SARS-CoV-2. So we can select that, and then based on the this nucleotide completeness. So if we are interested only in the complete sequences, then what you can select the different parameters. You can select a host if you are interested in the human samples, if you are interested in the vat. So you can select all these options and select those criterias. So then how you can download this data. So for example, we have selected this data and this sample and this sample. I'm just taking one these examples. So then you just need to go to the download. To download the sequencing data, that is FASTA, we can directly go to the next and then go to next and just download this. So you will select can download the only the FASTA file of all those sequences. So beside, if you're interested to get the information exactly what this table representing, and you need all this information of your selected sample, you need to select this CSV format. And for in this table, what you will obtain, let me just show you. So you can select exactly the information related to your sequences that you have already downloaded. So you can select its different any of these option and all the options and required sample that you are needed. So then you can just go to the download. So if I just show you, this is our FASTA file, FASTA sequencing file, and this is our file which contain the information related to these sequences. So here, this information we will obtain the other file which contain the FASTA, only the FASTA sequences. So just let me show you. So I'm opening this file just in this doc file. So this is the FASTA sequences. If you are familiar with this FASTA, you know that it is always its name and all the information start. First line is always that is started with a greater than sign. Then the followed by the sequence of that particular file. So you can obtain the data if you are interested in some viruses from this NCBI virus resource. So I'm not saving it. So is it clear to everyone? How we can obtain data from the public repositories, and then we can explore those data and perform any analysis using different tools when you have gone through these courses. Like if you are interested in already all of you must have gone through transcriptomics courses. For that, you know that you need a simple gene expression table. So we can obtain gene expression table from directly already, that is already processed data that is available under the supplementary file. Then if we want to generate and want to process our own data, so then we can download this NCVI run table and then run our RNA-seq pipeline to generate that gene expression table. So then we can perform various exploratory analysis. And if you are interested in specifically interested in like genomics analysis, for that you can obtain a data set from like you can obtain sequencing data. That is, if you are interested in the viruses, then you can go to the and directly go to the NCVI virus. So if it is clear to you, and even if you face any issue, or if you have any queries, still you can post on the forum. And meanwhile, you can submit your research project. So already some of you have already submitted their project. So when you are submitting your project, let me just go over this link. Okay, so let me just share my screen again. So here when you are submitting, right? So under the project summary, you also submit like what methodology that you are going to use. I mean, what methods that, for example, what tool, once you have that data, whatever the data that you have for your research project, and also under the project summary, also mention that exactly what you are interested in a particular, I mean, methodology that you are going to use. For example, once you have a process data, you want to perform PCA, you want to perform hierarchical clustering, or want to perform differential gene expression analysis. So those kind exact tools that you are going to use, you must mention those of your methodology as well. So if any of still some of you want to submit your research project, and so submit at this, already Ilya has shown you, this form is available. So under the forum, if you go, the first link, you will get this project form. So in the project form, you can submit your complete report, project title, project statement, and project summary. Then also provide the link of raw data, and if the publication is available, so also mention that. So thank you, everyone. Uh, you can still post your queries on the forum, and we will look into that. And thank you for joining today's session. So thank you, everyone. Okay, so we are looking forward for your projects. So submit your projects as soon as possible. So thank you, everyone.